Hello everyone. During this PowerPoint, we're going to specifically start looking at Activity 12 and the Hardy-Weinberg equation. My first question for you is, what exactly is the Hardy-Weinberg equation and why is it important? The Hardy-Weinberg equation allows us to make predictions about the specific genotype frequencies within a population. The Hardy-Weinberg equilibrium shows that there is no evolution happening within a specific population. For this equilibrium to occur, we have to have 5 conditions that must be met within that population. This includes having a very large population, no immigration, no natural selection, random mating, and no mutations. Do you think it is common for a population to have all 5 of these conditions? It is possible, but just not very common. Even though it's not very common for a population to meet all 5 of those conditions, it's still a great model for us to use as scientists. The Hardy-Weinberg equation allows us to compare data that can help us understand if a population is evolving. Here you can see the Hardy-Weinberg equation and what each letter represents. But first, I want to explain the relationship between those two equations you see on the slide. As you can see, if we start with the p plus q equals 1 equation and square both sides, we would FOIL and we would end up with the p squared plus 2pq plus q squared, which again is the Hardy-Weinberg equation. So now that we can see how these two equations are connected, and we can better understand the Hardy-Weinberg equation by looking at what makes up each of those equations. So P is the dominant allele. So for us, when we're talking about Activity 12, we could see this as a capital D. Q is the recessive allele, which when we're talking about Activity 12 would be the lowercase d. P squared is our homozygous dominant, meaning capital D for Activity 12. Q squared would be homozygous recessive, so 2 lowercase d's, which would also result in a light-colored mouse. And then lastly, 2pq is our heterozygous, so again, for Activity 12, we could view this as capital D, lowercase d. When using the Hardy-Weinberg equation, we usually will always start with the homozygous recessive, meaning q squared. But why is this? We do this because we know the genotype for homozygous recessive. So for example, we know that that light-colored mouse has 2 lowercase d alleles for its genotype. But we don't know that dark-colored mouse's genotype. That mouse could either be homozygous dominant, 2 capital D's, or heterozygous, meaning a capital D and a lowercase d. So since we always will know the homozygous recessive, it's a great place for us to start. You may have noticed that the Hardy-Weinberg equation always equals a 1 or 100%, and this is because it equals the total genetic component of that population. During class, we watched a short video on the Hardy-Weinberg equation. I will provide this link on Schoology. But at the end of the video, they presented this question for us to solve on our own. I will solve this question for us next. The question is, 12% of people in Ireland have red hair. What percent of this population is homozygous for non-red hair? So first we know that red hair is recessive. So for our non-red hair, that is going to be dominant, so a capital R. And our lowercase r will mean red hair. The problem is asking us for the homozygous dominant because it's the homozygous for non-red hair. So we know we could say that this is p squared is what we're solving for. P squared again would be capital R for us. So we are given that 12% of the people in Ireland have red hair. So that 12% can be turned into a decimal, so 0.12. And that is showing us our recessive, homozygous recessive, because we're dealing with red hair. So that's going to be our q squared. So now that we have q squared, we can find q. This can be done by just taking the square root of 0.12. So then our q would equal 0.346. So now we already have our q squared and our q. Next, let's use the equation p plus q is equal to 1. So to solve for p, we'll do 1 minus q. So 1 minus 0.346. So p would then equal 0.654. Lastly, we just have to find p squared now. So if we square that p value, we'll get our p squared of 0.428. And since it asks specifically for a percent, we can turn that into about 40, we'll say 43%. And this again shows the population that is homozygous dominant, meaning that they don't have red hair. Scientists will compare their data that was produced by using the Hardy-Weinberg equation. When comparing their data, if they find that they are the same, then the population is in equilibrium. But if their values are different, then the population will be considered evolving. When their values are different and a population is shown to be evolving, it could be for a variety of reasons. These reasons include mutation is happening, maybe natural selection, genetic drift, which basically means that a large population doesn't exist and it's a small population size, sexual selection, or gene flow, which means that there is some movement happening within the population. This slide shows us the stamps we will be collecting for Activity 12. This includes a stamp for our pre-lab prediction, for answering those book questions and pasting them into our notebook, by putting in our mouse data collection sheet into our notebook, by putting our class data into our notebook and graphing that class data, also by answering the AQs at the bottom of the class data sheet, and performing Hardy-Weinberg calculations for Generation 1 and your last generation and your class data. These calculations will help us better understand if evolution was happening within our mouse population.